Okay, very good afternoon to everyone here in Patancheru, and I think good morning to our other locations in Africa. And I believe that there are many colleagues they have been connected. So thanks a lot for coming to this important talk. And I can see the hall is full. And here the people are from genomics, molecular breeding, physiology, and from all the different disciplines and cell molecular biology or whatever you think. So this is fantastic. And we know that well why this is the so much crowd, because generally we don't have so many people, Gary. So but this is your name that all the people have come. So let me introduce very briefly about Gary, and everyone is well aware with Gary's name. So he is the senior program officer right now at Bill and Melinda Gates Foundation, and he is also the crop productivity lead within the R&D team at Gates Foundation, and he charged to coordinate the foundation's investments aiming to increase the rate of genetic gains delivered to smallholder farmers in sub-Saharan Africa and South Asia. And I always say, and we would like to give credit, full credit to Gary, because of Gary's reinvention or invention of the genetic gain word during last 3-4 years, we thought to name our program on the genetic gains. So, so this is one of the reasons. So thanks a lot, Gary, for helping us to name or rename our program. And Gary has been basically Canadian national, and he studied crop science at University of Guelph long back. And then, but he's having the experience to work. Okay, but anyway, so he got experience to work at different places, including SIP, and then he also joined Iowa State University, where I think you did your PhD work from Iowa, yeah. And then after graduating from there, he worked as a commercial flex breeder in Canada, and also taught plant breeding at Nova Scotia Agricultural College for 10 years. And then in months, he came back again to the CGIAR system and at IRRI, and he joined at IRRI in 2000, where he was working as upland rice and then rain-fed lowland rice breeder. And one of the most important contribution amongst many, that he established the drought tolerance trait pipeline at IRRI. And I think he was one of the pioneer, or probably pioneer, in not only in rice, across the other crops, that where they have identified the major QTLs for drought or yield under the drought stress, which became very popular. And many of you may be aware that Arvind Kumar, who was here, he was a postdoc with you, Gary. Yeah. So after that, he took this legacy from Gary, and then he has successfully utilized that QTL, drought QTL, in many breeding lines. And this is making great impact in the rice breeding in India and in many other countries. And after that, he joined CIMMYT in 2006 as a maize breeder, and then he became the technical breeding lead in 2009, coordinating efforts to optimize CIMMYT's maize pipeline to increase genetic gains. And I already mentioned that after CIMMYT, then he moved to the Gates Foundation. One thing is very clear, that throughout his career, he has combined practical cultivar development, and his programs have released several varieties in rice, maize, and flax. So basically, he always combined the practical work with the theoretical knowledge on and with an aim of maximizing the rates of genetic gain in stress-prone environments. And we are very lucky that Gary is with us. In fact, he came here to finalize some other project, and we have been working with him from morning to now. And I think you have to change something in your program. But anyway, let's see. He will be leaving tonight. But anyway, we are thankful to you, Gary, for coming here and also for helping us, our program at ICRISAT. And we are looking forward to hear this exciting talk from you. And this is about modernizing public plant breeding programs to deliver higher rates of genetic gain to farmers in the developing world. So please join me in welcoming Dr. Gary Atlin at ICRISAT.
Thank you very much, Rajiv. Where did you get that?
Okay, so you can.
Okay. Yeah, thanks a lot. No, I absolutely did not invent the term genetic gains. That's that's an old story, and I have a clicker. But we are at the Gates Foundation very concerned with helping public crop improvement programs increase their effectiveness. So it's a great pleasure for me to talk to you today about what we're doing and what you're doing, and I thank you for squeezing me into your busy schedule, because we decided to do this on fairly short notice. It was like a nice add-on for me to a day where we're essentially negotiating an agreement on genotyping services. So why does the Gates Foundation invest so much money? And it's invested a lot of money in the CGIAR and crop improvement in general. I guess through the CGIAR, it's invested something like $900 million over the last 7 or 8 years. And of course, we do this because we feel it's an important pathway to alleviating poverty by increasing yields. Increased yields result in increased food security and incomes for smallholders and lower prices for the non-farming poor. But in order to achieve these benefits, it's really important that we actually increase yields. So it's important that we track this in our investments in crop improvement. The way we're thinking about the problem is changing a little. We used to have a very strong focus on traits, and we're still very concerned with supporting effective trait pipelines. Our initial investment trait strategy was sort of to solve the problem of drought by breeding drought-tolerant varieties. I think we're a little more sophisticated now in that we're trying to help breeding pipelines deliver a steady stream of improved varieties. We're also very concerned with increasing both the rate of genetic gain and the rate of turnover of varieties, because by having varieties moving into farmers' fields that have been bred in the current conditions, it's really the most important mechanism for adaptation to climate change. The farmers who are best protected from climate change are those who are using varieties that were bred in the current climate. Just a few words about how our approach to investing in crop improvement is changing. It does look to me, this is not policy, this is my impression of what's happening within the foundation at the moment, that we will remain in crop improvement and R&D, but likely at a somewhat lower level than we have been. I don't, because there are increasing investment demands downstream of the research and breeding support that we've been providing. I also suspect we'll be making, within particular crop environment combinations, fewer grants, and grants that look more like major programmatic support, something like TL3 or HOPE. We had been making a number of relatively small grants on specific diseases, specific issues, specific traits. Not likely to do as much of that going forward. It'll be more support for integrative types of investment. And focusing on optimizing the rate of genetic gains delivered in farmers' fields and modernizing breeding and seed systems. In terms of access, there will be a strong focus on ensuring that women farmers have access to the products of research. It'll be very important to demonstrate that we've thought about the pathway to ensuring that women have access to new varieties and new tools. And strengthening national capacity will likely be as important a focus or more than direct products out of, for example, CGIAR programs. And we're really moving to a focus on metrics that give us a better handle on value for money in these investments related to genetic gain and adoption. So, for example, less emphasis on metrics like variety release, publications, more on the rate of genetic gain delivered and the rate of adoption of products. A few impressions about what donors want from the CG and from agricultural research for development. These views are diverse, but there, obviously there are serious concerns about governance, particularly the CG system at the moment, that are being worked through. And my feeling is that the funding problems that we're seeing right now are not going to go away quickly. I think there are concerns that business as usual will not attract new funds into the system. In terms of crop improvement, the CG is not really considered to be cutting edge by a number of the donors. So we're very anxious to help alleviate those concerns and help programs modernize. And our focus on delivering and documenting genetic gains from crop improvement investments, I think you'll see that USAID has a similar approach now. Other donors may have similar interests in breeding modernization. So my impression is that CG centers will need to position themselves not only as direct providers of products, but as the best providers of modern breeding approaches to national systems. We also think that CG needs to operate much more as a system, and that individual centers and programs may not have critical mass to deliver on the promise of genomics. So we've been trying to help stand up shared services like GOBI for genomics to try to help the programs really organize their genomics data in such a way that breeders can use it directly, provide tools like the integrated breeding platform for breeding informatics modernization. And because I think the funding situation is not going to improve a lot for a while, I think this is going to drive us to look for, consistently for increased efficiency and reduced costs. I think it's going to be difficult, but now is the time really to look at every single thing that we do and try to get as efficient as possible. And I think, the way to position ourselves for funding will be to modernize, to very clearly help national systems modernize, and to demonstrate our impact on productivity. So moving on to the changes in breeding, we've been focusing, and my career was primarily on trait-based approaches, delivering large effect alleles for stress tolerance like submergence tolerance, anaerobic germination, drought tolerance in rice. I'm sorry, I have a lot of rice photos because I was working in rice and maize, so you'll have to apologize. They're just there for illustrative purposes. But this type of approach won't be enough to deliver broad yield gains or climate change adaptation. Very important to do so, but we're finding that in a lot of systems, major genes for stress tolerance, for example, are not enough to drive widespread adoption. There will be some systems where stress is sort of the overriding factor, and in many of the crops that you're working in, it is. But in rice and maize, genes like submergence tolerance, sub-1, they're going to drive adoption in areas where stress is very frequent and very severe, but not, in terms of very broad areas. Because farmers, I think in this country, in many situations, income from cropping is now maybe less of the livelihood mix of many farmers. So as farmers become more commercialized, I think they often are more concerned with yields in the better years, than whether they get a complete loss or a small crop in the worst years. So, that orientation of farmers is changing. Doesn't mean we have to take our eye off the protective traits, but it means we also have to be very concerned with yield potential and quality in order to ensure that the products are adopted. And adoption really is an issue in many species. An example from rice. Many farmers in the developing world are using very, very old varieties. So there's just been a survey conducted by IRRI of average age of rice varieties in farmers' fields in eastern India in the wet season. So you've got the major varieties listed here in terms of the proportion of area under each of these varieties in the year of release. And doing that, we can estimate an average age of area weighted of varieties in farmers' fields. And it's about 28 years in India in the Kharif. So, farmers who are growing varieties that are on average 28 years old, and probably the cross was made 10 to 15 years before that, they are not, using varieties that were developed in the current climate. They are at risk. So we really need to be thinking about how do we make this system much faster, much more responsive. Now, in some commodities, especially in highly commercialized and hybrid crops, where there's competition among breeding programs and seed companies, the average age of varieties is much lower. In others, even in say in maize in Kenya, another system that I'm fairly familiar with, where hybrid maize penetration in Kenya is very high. Hybrids have been around for a long time, but the average age of hybrids in farmers' fields, because the industry is not competitive, the main company, Kenya Seed Co., has very little in the way of serious competition in the main maize-growing areas. Average age of the hybrids has been about 15 years for a long time, whereas in the U.S., average commercial life for maize hybrids is about 4 years. It's very similar for barley in the UK. And as I mentioned before, farmers who are using newer varieties are in a much better position in terms of adaptation to climate change. So we have to think about, in addition to generating gains more quickly, higher rates of gain, how do we, particularly in pure line crops, in crops where there is not a large commercial sector, how do we generate a faster rate of varietal turnover? Why are varieties hanging around so long in farmers' fields and in many pure line crops? I think, the way I think about it is that we really had you can think of three generations of varieties. In cereals in particular, where there was really a green revolution based on more responsive varieties. The first generation of green revolution varieties really sold themselves on the basis of very large visible differences in plant type. You don't need an experiment to tell you that IR8 is much higher yielding, than PETA. You can, a farmer can look at that and just see it. And then the second generation of green revolution varieties really sold themselves because the first generation was so terrible in terms of quality generally and in terms of many biotic stress problems that, the next generation of rice varieties, for example, IR36, IR64 solved a lot of those problems. And then we sort of got stuck. In many species where you did have this type of progression from traditional varieties to much more responsive, improved varieties, once the plant type was right, the quality package was okay, the biotic stress package was okay, those varieties have often stayed in the field for 30 years because the breeding methods we were using did not really improve much after that in terms of yield. Rates of genetic gain in staple grain crops have been fairly low. I think, generally speaking, too low to drive adoption where they've been measured. And they're not usually measured. I don't know how many of your programs have actually measured the rate of genetic gain in recent years. But it's very important that a breeding program do that you conduct, you regularly monitor your progress through formal IRRI experiments from time to time, but also work out a way to do it within your breeding pipelines. The rates of gain in better breeding programs, for example, in the Corn Belt over about 80 years have been documented to be around between 1 and 1.5% per year. They may be increasing now, at least in absolute terms, if not in percentage terms. By the way, I'm happy to leave this talk for anybody who wants it. And under optimal conditions, Simmet wheat, Simmet maize, which have very high selection pressure for yield, are generating rates of gain that are fairly similar. Once you get into tougher conditions, the wheat and maize programs have estimated rates of genetic gain recently. They are lower, well under 1%, 0.6 roughly. And their rates of genetic gain estimated from the IRRI irrigated program, which is the only rice program I'm aware of in Asia that's actually done this, have actually been very low. More about that later. And these are gains measured in research plots. It's likely that gains in farmers' fields are lower. Have any of the groups done an IRRI study, estimated rates of genetic gain in the ICRISAT mandate crops recently?
We did this thing in pearl millet, Rakesh. You have idea about that? Yeah. So
Yeah. Oh, if you could provide me with the reference, that would be terrific.
Yeah, I can give you a specific reference.
Okay, great. I'd appreciate it. Yeah.
Another crop we discussed, and I think that we will be having soon some data.
Yeah. It's remarkable how rarely it's done. I just wanted to run through, the way I sort of think about how breeding programs are organized. The core engine of any breeding process is a rapid cycle process where you intermate their best lines, generate new populations, select superior lines, and as soon as possible you reuse them as parents. And then of course we have a trait pipeline, that may go from a discovery unit through to a forward genetics program that identifies QTLs and either integresses large effect IBD segments into lines or comes up with some type of genomic prediction haplotype that's used in selection. And this is the engine of breeding that really drives gains quickly. So we really have to look at our programs and say, how fast is that engine working? How quickly are we cycling through that process? How effectively? And then we need to select candidate cultivars that fit a product profile. And a lot of the programs that public sector programs don't have a very formalized product profile. The breeder has an idea about what farmers want, what the market wants, but very often that's not really documented very well. It's not tested. It's not validated with end users, and so we really think that developing very formal product profiles and breeding targets is something that the programs that we support should be doing. And then from these candidate cultivars, national systems will identify and release superior replacements for current varieties. And I've got the word data there. You probably can't see it, but highlighted with exclamation points. One problem of a lot of public programs is they don't really generate enough data to be able to go to the Ministry of Agriculture, to the extension system, to companies and say, this product should replace what you're growing now. We have to have very high quality data and large quantities of reliable data to be able to make that recommendation. And it's, whose job it is? It's the breeding organization's job, because nobody else is going to generate that data. You know, nobody else is going to have enough information. So we really need to make sure that throughout this process we have the data at the end to be able to make these recommendations. I'm going to have to speed up. And then, we need to be, these arrows should sort of be pulsating to indicate that, there's a steady stream of these new products coming to companies and to farmers. And we're very concerned with releasing varieties. We don't think very much about replacing old varieties. Multinational seed companies think about their product mix, their product life cycle, and they not only introduce new varieties, they withdraw old varieties. And withdrawing old varieties is quite difficult for the public system to do. The Indian public system is now using some policy levers to increase rates of varietal turnover. For instance, I believe now there are regulations indicating that varieties that are more than 10 years old can't attract seed subsidies. They're not eligible for a subsidy program. Good breeding systems generate rates of genetic gain of 1 to 2% per year. That's what you can get if you're working very effectively in farmers' fields. Most programs much less than that. A few key lessons that we've learned from our investments are that yield potential, quality, and maturity seem to be driving in many systems the demand for new varieties. Stress tolerance is extremely important. It's what I've worked on most of my career, but it alone will not drive adoption. I've mentioned that already. We've also learned that farmer-to-farmer spread of varieties happens very slowly. It's not enough to drive adoption. That breeding organizations and seed systems really actively have to push varieties. And that, as I mentioned, most public breeding and many private breeding organizations don't generate enough data to be very confident in their recommendations. And we're seeing that small farmers are buying seed in many crops that they traditionally didn't buy seed in. Replacement rates, even for pure line crops, are higher. So we're really thinking about seed companies as the main pathway into farmers' fields. And the key metrics we're using for our crop improvement and seed systems innovations are rate of genetic gains, of course, under farmer management, and that, it's very hard to measure in any relatively compressed period of time rate of genetic gain. You need about 10 or 12 years of variety releases anyways, a couple of cycles of variety releases to measure your progress. So we're very concerned with intermediate metrics of the effectiveness of a breeding program that are related to genetic gain, like selection intensity, population size, heritability, extent of your testing system. And we've incorporated a lot of these metrics into something called the Breeding Program Assessment Tool, which is a mechanism for an instrument that breeders, research managers can use to evaluate their programs and see where they are stronger, where they are weaker, where they can be improved. And I believe ICRISAT has used this for some of its programs in Africa already, and will be applying it to more of its programs in South Asia and at headquarters. And of course, as I mentioned, average area weighted age of varieties in farmers' fields is really the key metric for the effectiveness of a seed system. What are the routes to increase genetic gains? I'm going to focus now on genetic gains. Obviously, these are basically what I'm listing here are the components of the breeder's equation, written out in words. Bigger programs or higher selection intensity for yield. And how do we get there? We get there by mechanization, automation, and digitization. And I was really, I met with a number of breeders and the farm managers yesterday, and I was impressed by the level of support that breeding teams at ICRISAT have for mechanization, in particular, of field operations in breeding. Breeding programs need better, more genetic variability, elite exotic materials. They need to increase the heritability of their screens, either through or both through higher quality phenotyping and more replications and marker-assisted selection. A very important issue is to accelerate breeding cycles. Most public sector breeding programs recycle promising lines into their crossing blocks too slowly. Management in many breeding organizations really doesn't understand very well how product development programs should be organized. We tend to think of plant breeding as research. It's not research. It's product development. It's much more related to, more similar to engineering than to basic research, in the sense that we take well understood scientific principles, we use them, we optimize pipelines with them to generate and deliver products. We do some research, of course. And we need well-trained staff who understand product development. And a lot of breeders are going to need to learn a lot to work in product development teams. Plant breeding is getting quite complicated. We have a number of tools that are really ready to be used now that breeders need to be supported on. It's not going to be an individual activity. It's a team sport. Breeding programs at this point really need to be redesigned to exploit low-cost diagnostic markers. We've invested hundreds of millions of dollars across the CG system and other research systems in genetic analysis over the last 20 years or so. It's really time to deliver on that investment. We've developed many diagnostic markers for important traits, and they're currently being used for backcrossing only, not forward breeding, which I understand now is also some type of program name here. So I'm sorry about that. But there's really no reason for us not to start using diagnostic markers very early in the program. The cost of SNP genotyping through commercial services like LGC, Intertech, we're negotiating right now, is less than 20 cents per marker. And DNA extraction is less than $1 a plant. So we expect to be able to provide the breeding programs at ICRISAT and at other CG centers and in the national systems the ability to genotype a line for anywhere between 1 and 10 markers for well under $2 very shortly, within the year. And that really means that we have to, and ICRISAT is leading a project to put this into place. At that price, you know, if you have good diagnostic markers, $2 a plant, that's cheaper than virtually any phenotyping that you can do. So we really need to think about how to reorganize our breeding programs to take advantage to this. It means that lines can be fixed for key diagnostic markers before they're subjected to expensive phenotyping in the field. This has been on the horizon for many years. It's here now. So it means we have to change the way we operate. But the ability of a service provider to offer these costs depends on the volume and coordination with clients. They can't in order to get these prices, we're going to have to be very careful about how we extract DNA, how we provide the samples. We're going to need to use very standardized processes. So how do we have to look at reorganizing programs to use these tools to increase selection intensity for yield? And that's really where we've been falling down. A lot of breeding programs, particularly in pure line crops, have been applying very low selection intensities for yield. They've been focusing on visual selection in pedigree programs. So what would you do if you had even as few as 1 or 2 diagnostic markers that you could assay, all costs included, for $1 a plant, $1.50 a plant? Well, I think what you would do is what many commercial programs have done, the commercial soy programs in the U.S., for example, canola programs. You would develop as many fixed lines as possible at minimum cost per line, using some variant of single seed descent, or in some cases, if you've got very cheap doubled haploid systems, either in the field or in the greenhouse. Pedigree breeding is really too expensive and too ineffective after you've gone through your first and second stages of your green revolution process. You've got the plant type right, easy to select that visually. You've got the basic package of quality and disease-resistant traits. Then you would conduct low-cost and high-throughput screens for must-have quality traits. You might do that with NIR. Disease resistance with low-cost SNPs, and plant type, maybe a single row or a small plot in the field. And then you would very quickly move to multi-location trials. You would take the survivors of that initial screen and put them in unreplicated trials at 4, 5, 6 locations and get a yield estimate at a heritability of 0.5 or higher. So you would need, usually you'd need 4 or 5 locations to do that. You would use managed stress screens also to ensure that you were eliminating material that was very sensitive, as long as you were sure that the correlation with performance in the target population of environments was high and that the repeatability, the heritability of the screen was high. Making sure that you didn't, select on traits that are very low in heritability and screens that are low in heritability. So the IRRI breeding program has really gone through a process of reorganizing itself in this way. So I just wanted to run through with you what they've done. With our support, and what drove this was their recent IRRI experiments. They published an IRRI experiment in the late 1990s, which estimated rather low rates of genetic gain, and these were confirmed again in 2014, 2015. In the wet season, the annual rate of gain was about 0.67%. This is likely an underestimate, an overestimate of this rate of gain. I'll explain why in a second. And in the dry season, the rate of gain is only 0.15%. And that, again, may be an underestimate, because I don't know if you can see this, but IR8, you know how you do an IRRI experiment. You take, these are about all the major lines that have been introduced, released by IRRI from IR8 in 1967, 1968, through to lines that were released in 2013. IR8 yielded in these trials, 2 seasons in the dry season, about 7.2 tons per hectare. But we know that in the late 60s, IR8 was yielding much more than that at Los Baños. They knew how to measure yield in the late 60s. It wasn't, they already knew how to do that. It was yielding about 10 tons per hectare. So what we may have been seeing, even this low rate of gain of 0.15%, may have been maintenance breeding. So as a result of these low rates of gain, the program was, the pedigree program was scrapped in 2013, which was a big step. It was the biggest rice pedigree program, I think, in the world, and redesigned from scratch. Line development was accelerated, and its cost greatly reduced by moving to single seed descent in a rapid generation advance, they call it RGA system. Selection pressure for yield was greatly increased by expanding multi-environment testing and introducing it at a much earlier stage. And the breeding cycle was shortened by aggressively moving promising new lines back into the crossing block after relatively short periods of testing. They also developed an integrated breeding informatics system, B4R, breeding for rice. You have been leaders in implementing the breeding management system. Field data collection is completely electronic, and they've done what you've already done, which is mechanized most aspects of planting and harvesting. So this is what the pedigree program looked like up to about 2013. It was very traditional. 2 generations a year. They planted out in one trial at Los Baños, started about 400,000 seedlings every year in the F2, selected visually through the F7 derived F8. In the observational yield trial, we put out at only one location 2 reps, 600 or 800 lines, move something like 500 of those forward into 3 replicates, and then only about 30 of them would go into multi-location trials. And that's really where your selection pressure for yield starts. So this selection intensity for yield was very low. So the new system generated lines via single seed descent, and they control photoperiod to speed things up. So it's done in a screenhouse. And so they get 4 generations or 5 generations in 2 years. So they were able to get to the F5 derived F6. And then they move right in the 3rd year into unreplicated testing, 1,400 lines, but replicated across locations. So they get a multi-location yield. And the best lines coming out of that are moved back into hybridization, because in fact, you get a lot of your progress for yield from your first yield trial, if it's a good multi-location yield trial. And then, of course, they line out and move larger populations into multi-location testing. And they're using their genotyping lines, these lines going into replicated testing and beginning to use genomic predictions. They also, of course, use managed stress testing, particularly in the rain-fed programs. They replaced pedigree breeding with indoor single seed descent, got 3-4 generations a year, and this reduced line development costs to about $5 a line. The breeding cycle was reduced from about 9 years, 8 or 9 years to 3. About a 10-fold increase in selection intensity for yield. They haven't introduced diagnostic markers yet on a big scale, but you can take any one of these plants and sample them and select and cut the populations going into the field that way. There'll be heavy use of NIR at the early stages for quality parameters as well. And the survivors can be profiled using a high-density genotype before multi-environment testing. And start building up, prediction models. The program has also introduced very formalized product profiles that breeders are aiming at. So a corporate decision is made on exactly what are the product streams that are being produced, so that, basically we decide what variety we want to replace. The question came up the other day, somebody asked me, how do we think ICRISAT is doing relative to other centers? I think it's doing pretty well. I mean, I was just thinking about a number of areas that, in terms of modernization of its programs, where different major programs are. And I know ICRISAT deals with a lot of crops, and there's variations among those crops. But in terms of mechanization of breeding, in really good shape, integrated data management, ICRISAT and IRRI are definitely farthest along. CIMMYT is following up. Outsourcing genotyping, CIMMYT's outsourced all its genotyping. ICRISAT is moving in that direction and has a lot of good in-house capacity. In terms of optimizing breeding pipelines, I think that starting formal product profiles, I don't know if you have them. Some of these are blank because I don't know. So formal advancement systems, most of the centers don't have them. CIMMYT and IRRI are introducing them. Measuring rate of genetic gain, you are in some crops, so I'll stick something in there. Georeferencing the farm, almost nobody's done that. So we're, in some areas, moving along well. So what is the Gates Foundation doing in terms of trying to help the CG and NARES breeding programs modernize? We're concerned that relatively few centers or national programs have the knowledge or management skill in-house to drive major breeding program reorganization. And this also involves changing teams, changing incentives for breeders. The changes required are both managerial and technical. There's a lot of technology and management changes on the horizon, including the application of engineering type methods to plant breeding, operations research. And in general, I think breeding teams are going to need strong consultancy support for program modernization. The CG should be well positioned to provide this consultancy support, but it doesn't, a lot of the programs don't work easily across crops. I think as a multi-crop center, ICRISAT is in a good position to sort of pool best practice across its crops and help national systems on an institutional level raise their capacity. What are the support systems that are missing that we're trying to help with? Well, product profile development, really using market research. We used to talk a lot about participatory varietal selection, participatory breeding. Why do we need it? We need it because we don't do good market research to really define, understand what farmers need, what millers need, what consumers prioritize. ICRISAT is in a good position, but many programs aren't in terms of mechanization and automation of breeding operations to increase throughput. Detailed support for pipeline optimization. So this shift from pedigree breeding to more effective methods of breeding needs to sort of be penciled out in terms of each of the steps, and it's good to get some help with that. Formal advancement processes where you bring in the institution really stands behind a particular product, whether we're going to market it or not. Dissemination support, a number of our programs. The traditional paradigm was we said, well, this is the problem with the national system. We've got a good product here, we'll describe it, somebody else's job to move it out. And that is not driving varietal turnover. The breeding organization doesn't have all the tools in its hand, but it has to make the dissemination plan. And then implementation of improved metrics, accountability systems, so measuring rate of genetic gain, uptake. We really need for the system a single, there's a lot of technology coming from the multinational seed companies, and it's very difficult for them to work with individual programs, individual centers. If we had a one single window for them to help us with things like seed chipping, genomic selection, imaging systems for phenotyping, I think innovation would move faster through the system. We've been trying to help stand up some centralized support systems like the integrated breeding platform, which you are using here, the breeding management system, I should say. You know all about it, so I won't go into any detail about that. You're probably familiar with the GOBI project, which will help organize the vast quantities of genomic data that we've generated. You've got almost 3,000 chickpea lines that have been resequenced. So what are we going to do with that? What is it? 300 billion? 3 trillion data points. So the GOBI project is going to help build that scaffold, which will allow us to relate the reference genome in the species to resequenced key ancestors. I know it's cheaper than $2,000 here. To parents that are profiled via GBS for maybe $30 a line, through to selection candidates that are profiled with low-density panels, but that will allow us to impute essentially the whole app map onto selection candidates. We're putting in place high throughput genome profiling systems in Africa through a collaboration between an Australian company, DART, and Becca. And of course, ICRISAT is leading on a contract to provide high throughput genotyping for diagnostic markers here. I've already, okay. We've been standing up these services, this last point here. at Gates, and it's very important that the CGIAR take responsibility in a corporate way for these systems. So the you know, we found that although, you know, we will not be able to continually invest in maintaining the breeding management system or maintaining Gobi. The system itself will benefit greatly from looking at, from collaborating across centers, sharing capacity across centers. But there hasn't been a mechanism for doing this. There's a proposal right now being developed through the current CRP process to develop a genetic gain support platform, which will help handle these services, which will help provide consultancy for optimizing programs, promote best practice and sharing services across programs, and link aggressively with multinational seed companies to license technology and help it be applied in breeding programs supporting the developing world. I think I'm going to have to summarize now. Sorry. There really will be need for stronger support for national program modernization, and it's support at the institutional level. This is going to be a high priority for donors. And I really think that institutions like ICRISAT are in a good position to provide this type of support because you work across so many crops. So you need to be thinking about how to do this. When you go to you know, try to help you go to a minister of agriculture in a country and say, we would like to help you improve your research system in maize or in millet. You know, that's not really the way to approach it. We have to approach them at the National Institute level. They need to share capacity in terms of biometrics, genotyping capacity across crops. And an organization like ICRISAT should be in a good position to help with that. I'll skip through to this. So in summary, as a partner, we've invested about $700 million in crop improvement in the CGIAR. We've already spent or committed about $65 million on shared services like the IBP, like Gobi, like the high throughput genotyping facility that Rajiv is leading on here. About $21 million in NARES and CGIAR breeding program upgrades, in addition to commodity programs like HOPE, like TL3. And we have about $10 million in the pipeline for some type of genetic gain support platform for the CGIAR and partners, likely in 2017 in tandem with or in support of the platform proposal going to the consortium now. And we will continue to help our partners design and seek resources for an international crop improvement system that really is more effective in delivering higher productivity and alleviating poverty. Thanks very much. Too long as usual, sorry.
Thanks a lot, Gary, for providing an outline for the different breeding programs and how we can maximize this genetic gain. And many of our breeding programs, they are aligned what you are proposing, but this is also a good suggestion and guidance for us that we need to move in this direction. So thank you very much for that. I think we can have a few questions if you are some suggestions or something. Yeah, so floor is open. And if people are following this thing virtually, they can also send some question if is there. Okay, some Ashok. Yeah. Ashok is our sorghum breeder.
So nice talk, Gary. The point is, when you embrace these methodologies, what is your take, like whether we increase the per se performance or reduce the time actually? What's the thing where we really gain?
You need to increase rate of gain per unit of time. So that's what the outcome we're looking for is. And to do that, you have to speed up your cycle, and you also probably have to apply higher selection intensity for yield. And that means, generally speaking, trying to generate lines as cheaply as possible, cut them down with the cheapest methods you can, cull your population with the cheapest methods you can. And what's happening now is that genotyping, after 20 years of promise, from the molecular side, is now actually cheap. If you can get DNA extracted, get 10 markers that are diagnostic, assuming you have them in your species, for $1.50, then that's really what you should do first. And that allows, what does that allow you to do? It allows you to apply higher selection intensity for yield and run much lower risk of spending hundreds of dollars and a number of years on a line, evaluating a line that you're going to have to throw out because of some basic defect in quality and disease resistance. Complicated problem, and it's scientists can advocate till they're blue in the face. This is a political issue. I mean, at Gates we struggle with this too. Obviously, we're strong supporter of providing transgenic technologies that work for farmers, allowing them to farm, and we have not come up with the magic wand that will allow us to speed up deregulation and deal with the political problems associated with this issue. It's very difficult.
Thank you. Anthony?
For several years there's been talk about using crop models to identify traits for breeding. It's probably been 10 years now, and people like Jim Jones and Graham Hammer do talk about it. Have you seen that come through in reality yet in your work?
I'm not, I've seen very impressive results coming out of Vincent's program. They certainly give guidelines to how both how traits can be identified and how they can be deployed. Yeah. It's up to the research programs themselves to determine how to weight that type of information in planning directions. So, the likelihood, say, of the Gates Foundation mounting a large crop modeling project is low. But if within a particular commodity, it was determined that this was a promising approach, then that would certainly be something that you could incorporate in a major breeding initiative.
Okay, we will take two more questions. Okay, Vincent and then Bindu. Yeah.
Was maybe related to this. I mean, what is the plan of the Gates Foundation to invest into basically the engineering of phenotyping? I mean, equipping a station with good old top-notch irrigation systems in representative locations, which would be determined by modeling, for example, just to produce better data. What is your is the Gates Foundation planning to invest into that?
I wish I knew exactly what the Gates Foundation was planning to invest in. We are advocating strongly for a modernization of the system, and we will invest to the level of our ability in. But there are many competing demands, and our focus has shifted substantially downstream. There's no doubt about it. We will continue to support crop improvement. I would very much like to see a reinvestment in the system. Whether we're going to be successful in putting together a coalition of donors to allow it to happen, I don't know. I would hope that we could work with non-traditional sources like the Green Climate Fund to make this reinvestment. But what you're describing needs to happen. I support it. Whether we'll be able to do it on a large scale is another story. We are supporting work on modernizing phenotyping, on using imaging, starting to apply drones, tractor-mounted imaging systems. Absolutely, we support it. And what I would hope to be able to put together as a next round of crop improvement projects, I think rather than TL4, or Hope 3, we're likely to be luckier or more successful in funding something like transforming chickpea breeding or transforming legume breeding, transforming tropical cereal breeding, in which we can invest in the type of improved phenotyping systems that you described. Absolutely, there is no point in doing genetic analysis on poor phenotypes. That's absolutely understood. Absolutely. The question is, who has the comparative advantage to doing that? Should the CGIAR get into genome editing in a big way? It's not clear. The comparative advantage of the CGIAR, it seems to me, is really at the nexus of having the populations, understanding the environments, understanding the cropping systems. It may be that editing alleles to feed into that pipeline, to package them into varieties and deliver them to farmers effectively, is best done elsewhere. I'm agnostic on this. My colleague who manages the discovery component of the Gates program, Vipula Shukla, doesn't see that the CG is probably the best place to do that. But there are different opinions around, I know.
Every time I say last and people raise their hand, so maybe this is the last now. Because Gary's schedule is very busy, so that's the
Yes, I know.
Oh, well, so's yours, I'm sure.
Nice presentation indeed, Gary. Your presentation on genetic gains is really very interesting. But here in ICRISAT, we have a paradox. We have crops which are self-pollinated. We have crops which are cross-pollinated. We have crops where you have these varieties. We have crops where you have single cross hybrids. So this term genetic gain, what I understand is you go on increasing the perceived performance of line every generation, right? So this makes a lot of sense in self-pollinated crops like chickpea, often cross-pollinated crops to some extent like pigeon pea, groundnut. But when it comes to hybrids, when it comes to crops where hybrids are prevalent, like pearl millet and sorghum, this does not complete the jigsaw puzzle because perceived performance of the line is not going to tell you everything as far as
No. This genetic gains estimate is, Duvik's classic IRRI study here. That's not on performance of lines. That's on hybrids. This estimate here from CIMMYT, I don't know where it is now, but yeah. That's on hybrids, not on you estimate genetic gain on the product. There we go. That's on hybrids.
I get your point, Gary, but here at ICRISAT, we are not supposed to produce hybrids. We only produce hybrid parent lines.
But you test them, presumably you test cross them to something while you're producing them.
We just share our hybrid parent lines with our nuts partner.
But how do you select them in a test cross?
We select on the basis of perceived performance. That's it.
How do you know it's a good hybrid parent if you select it only on...
But in those cases, when you listen back. So in those cases, when you give these things and your partner they develop the hybrid, then you should have the data. And
that's right.
Yes.
So this is not that.
I have to say that a program that's releasing parents for hybrids that doesn't test those parents in hybrid combination, that sounds very strange to me. You must be testing at some level.
on limited scale, maybe.
Okay, good. So as I said, we already are cancelled one meeting what was planned, and I don't know that whether we will be cancelling the other meeting because we have to have the discussion with DG also, and we have to finish one other program, so he has to leave tonight. So because of all these reasons, we have to close the session now, but this was a very interesting session. And as I mentioned earlier, so after the reorganization and in the genetic gains research program, this is the first talk from Gary and the two in the genetic gains. So we are very much privileged to have you, Gary, here and to give the guidance to ICRISAT breeding program. And once again, and I would like to acknowledge on behalf of the Institute, Director General and Deputy Director General Research, who could not be here, but they conveyed their greetings and thanks to you in particular, but in Gates Foundation in general, for extending the support to ICRISAT breeding program and ICRISAT in general. So thank you very much with these words. Thanks a lot, Gary.